Assalamualaikum, and our group will explain about two concerning biosafety issues and its mitigation methods. This group consists of myself, INTAN NURFARZANA, AFIAH, and NADIA FAUZI, each with their crucial roles in this video. Let's go to the first biosafety issues concerning synthetic biology, and that is bioterrorism.
Misuse of synthetic biological technique for terrorism is the first issue. By creating living viruses, researchers may better understand how the infection spread and develop specialized vaccines and medication to combat possible outbreaks. Bioterrorism can bring serious consequences such as epidemic illness, a threat of anthrax through the mail like in the United States. A slight mutation on the genetic sequences may result in a mutant virus with increased virus latency. Engineered bacteria by synthetic biology is currently a great concern as it brings harm just like the quote from Emit Ray.
Now we know about bioterrorism, let's see how to mitigate the problem. So, in the field of biological weapons, there is almost no prospect of detecting a pathogen until it has been used in an attack by Barton Gellman. So, in order to fight this problem, we must take proactive actions and not reactive actions when it's already too late. First off, enforcing and create laws. As you can see from the slide, these are the laws created to fight bioterrorism. The recent one being the largest and most complex, which was the anthrax attack in 2001, where 22 high-ranking officials were infected and 5 of them died. This led to the formation of a plethora of laws, which we have seen in the previous slide. Sufficient containment efforts must be done where two important characteristics must be taken into consideration, and that is definitive care and transport to the definitive care. Safety equipment must be provided and worn to work with bioweapon products. Sufficient laboratory practice, PPE, and closed containers, and other controls designed to remove or minimize exposures to hazardous biological materials are all factors that needs to be taken into consideration when dealing with dangerous materials. Last but not least, imposing genetic safeguards in the case of an outbreak. Three main characteristics surround this way of mitigating bioterrorism, and that is low escape frequency, which will prevent the rise of mutants escaping defined media and limit their growth in the wild. Second, robustness, where robust safeguards retain wild type levels of fitness while also maintaining containment in diverse growth conditions. And lastly, a modular safeguard, where it will allow many different strategies to be combined in one strain, enabling multi-layered safeguards, or for those safeguards to be transferred to different organisms, enabling portability. Now, one such way of this method is the hawk toxin gene. This gene will be inserted into a vector, such as E. coli, and expresses a certain type of toxin that will function as a deterrent to the rogue synthetic biology product. Moreover, this hawk toxin gene can kill a variety of bacterium types, easy to insert, and requiring minimal nutrition to express its toxin, satisfying the three characteristics mentioned before.
Few biosafety issues were extracted from an article Biosafety Consideration of Synthetic Biology in the International Genetically Engineered Machine IGEM competition reported by Guan et al. 2013. There are many safety issues raised regard to the IGEM competition throughout year 2008 to 2011 mainly concerning the materials and biobricks used in the projects. The team have identified that material used pose risk to the safety and health of the team member and others in the laboratory which involve physical and chemical risks. Also, some other projects from other teams have identified issues involving environmental risks. According to the authors, there are several biosafety issues that need to be resolved right away since more and more people outside of the established scientific community will start to develop self-replicating organisms. The advent of so-called amateur biology, also known as biohacker community, a group that has started to execute biological projects in their own garages and in homegrown molecular biology lab, has made situation even more dire.
There are some ideas suggested in the article to mitigate these issues. First, laboratory practices for synthetic DNA containing biobricks must be below biosafety level 2. Second, to reduce the risk resulted from material use, avoid the manipulation of any infectious bacteria and use only purified DNA as the chassis. Last but not least, for environmental safety, integrate the suicide circuits into the engineered bacteria.